2-minute neuroscience.
It's okay. You can keep doing what you're doing.
2-minute neuroscience.
What do you think I studied? I put 2-minute neuroscience on like 1.5 speed.
How's that go?
Right there. I was going extra. I was confused that or did this we bought it since you're just having the 20% sale on anything during Fridays.
Damn, okay.
Okay, like, I think it's like 1 You should take all your test questions from there. Not the word. Behind the 2-minute neuroscience merge, so I think it's.
These are cool.
But probably don't put it on
Yeah. They rock, the brains are cool.
Yeah.
Yeah. Anyway, that's not 2 minutes, that's 8 minutes.
My like partner Ellie has her own girl, she's dressing for squares on her group, she's never singing. And she felt really embarrassed wearing this one. So I'm like, I'm just, what do you always do all this stuff? So I'm in an orange jumpsuit. Oh, yeah. Yeah. Oh, yeah. And I don't know if she's gonna survive right now, because I feel embarrassed with the great green cell color.
I just got-
Like, oh, cute. It's giving coordinated. Yeah. The dial is what makes cool. Yeah.
There we go.
I did go back and like do like spiral tire because it's like that. Yeah. You took out of my closet. All three. For some reason,
How do you exit?
Rating, I'm like laser. No, pretty much, I'm pretty much. Flash blood. Yeah, because I was like, and also, how did it end up in my suitcase to go? That's my better question. Yeah, like you would look at that, you're like, nah, I need that. It was really, I did it about work. I feel like making the mind go out, and now I just know about any clothes that I'm using all right. Yeah, it's great. That was like my line.
Oh.
And I wear this thing all there. No, I do.
Welcome to 2-minute neuroscience.
Ooh. There's no 2-minute neuroscience happening. Okay. This side's cooler. I don't know. You can still sit over there. It's okay. No.
by myself. Real.
Okay, let's go ahead and get started for today. How'd the movie go? Great. Big thumbs up there. Awesome. Okay, let's see. It's Thursday. How are you guys doing? Good? Okay, so where we're at, where we're going. Today's a kind of our last day of material prior to moving into our next exam, which is next Thursday. Okay, so what we'll be doing today is finishing up our slideshow. Oof, we'll see how far we get today, but however far we finish, that's what you'll be responsible for knowing for this third exam. I do have your questions that you turned in on Tuesday, which is great, but I actually think I'm going to hold off on those and actually maybe create some slides and answer them on Tuesday. Tuesday, we'll be spending our time doing reviews, going over kind of short answer questions for the exam. There will also be a Kahoot involved for self-assessment purposes. So hopefully I'll give you lots of opportunities to kind of identify what you know and what might still be kind of a struggle for you moving into this exam. A lot of this exam is focused on things like ion channels, action potentials. We've talked about NERTs and Goldman equation. And all of that's just kind of building up to the point that we're at today, which is discussing things like synaptic transmission. Before I get into it, one person did have a question as to like if I accept late work for like the in-class activities. Yep, just keep turning stuff in. 9.995 times out of 10, I will not take off any points. I set due dates just because if I don't, people will procrastinate till the absolute end of the semester. So I understand that inclination. But if the closer we can stick to due dates is awesome. We are a good portion of the way through the semester. And I will say, if you have not even thought about our end of semester project, talk with any of your folks that you're working with that project on. Now might be a good time to just kind of revisit the rubric and instructions for that end of semester project. Okay. Maybe that will be something also that we talk about on Tuesday, and I can give you guys a little bit more like structure related to that, talk about some possible examples. But I just want to make sure it's something that you do not forget. And then the end of November, you're at home trying to enjoy Thanksgiving and you're like, I have to do this project. Nobody wants that to happen. So maybe we could even set aside some class time for you all to kind of work together on coming up with some possible ideas. Okay, so with the upcoming exam, that reminder of, again, procrastination, I totally understand that concept. But the other thing is with the second exam, you will also need to turn in your study guides. So if you have not been working away at your study guides, now is also a great time to start working through those because those will be due next Thursday as well. If you have been procrastinating them, that's fine. It might just be another way for you to study and look at this material fresh as you're getting prepared for the exam. Cool? Okay. Yes. I do not care. I'll be honest with you. Like you can have one person. I think 5 is probably getting to like the maximum possible number. I think somewhere between that like 2 to 3 is kind of the sweet spot. But yeah, I think as long as you can have a group that is of an appropriate size where everybody can still contribute and do a certain amount of work. If you are a lone dog and you just want to do it all yourself, that's fine. I know group projects aren't everybody's jam. But if that's the case, then let me know and make sure you sign up on the sign up sheet just so I can make sure I'm setting aside the appropriate number of days for everyone to present, if that makes sense. So once you do have your group together, there is a sign up sheet on our Canvas page. Just go ahead and throw your name, and I think your topic might be asked for there just in case. So yeah, like I said, maybe on Tuesday I'll set aside 5 to 10 minutes for you to kind of identify your group, maybe come up with a possible project. And then maybe a few weeks later, we can kind of go through some kind of steps for planning for said final project. Good question, though. Any other questions? Concerns? We're good? Okay. Yeah, so I think we're going to kick off. Last time we're a few slides after this particular point, talking about synaptic transmission. And this video on myelin, which is an important part of synaptic transmission, was embedded, but it wasn't playing. So I decided to go back and fix that video. So we're going to kick off by watching said video and then kind of go from there. Sorry, we're just going to have to use my computer. Hopefully it works.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss myelin. Myelin is an insulating layer that surrounds the axons of neurons. Composed primarily of lipids, myelin helps to prevent action potentials, which are the electrical signals that travel along axons, from decaying due to electrical current leaking out through the axonal membrane. Myelinated axons thus conduct action potentials more quickly and efficiently than unmyelinated axons, and because of this, many neurons in the nervous system are myelinated. Myelin does not cover an axon fully. Instead, there are intermittent gaps in the myelin where the axon is exposed to extracellular space. These gaps are called nodes of Ranvier, and the sections of myelin that are adjacent to the nodes of Ranvier are called internodes. The nodes of Ranvier are rich in sodium channels, which open in response to an action potential traveling down an axon, allowing positive sodium ions to rush in. This influx of sodium ions rejuvenates the action potential and helps to prevent it from dying out as it proceeds along the axon. Because the nodes of Ranvier are not myelinated, however, the action potential slows down at each node and speeds up as it travels along the myelinated internodes. This gives the appearance that an action potential is jumping from node to node, which is known as saltatory conduction. Myelin is formed by glial cells, but the particular type of glial cell responsible for myelinating axons is different in the peripheral and central nervous systems. In the peripheral nervous system, glial cells called Schwann cells form myelin. Each Schwann cell wraps around one segment of an axon many times to form one internode. In the central nervous system, oligodendrocytes form myelin. One oligodendrocyte can produce dozens of internodes on multiple axons. Because myelin is white, myelinated axons appear white and make up what is known as the white matter of the brain. We can see the importance of myelin to healthy neural functioning when we look at diseases where myelination is disrupted. In multiple sclerosis, for example, deficiencies in myelin disrupt neuronal communication, resulting in a variety of physical and psychiatric symptoms.
So, yeah, I feel like that's a
Welcome
really
to
good synopsis about myelin and the importance of myelination. We just talked about, again, action potential generation and how that action potential travels down the axon. But we kind of, for the most part, kind of ignored the myelination status there. Whenever I describe myelination to somebody who maybe isn't completely familiar with it, I always think about the electrical cords that we use to connect different devices and how we have this insulating factor here that stops the electrical current from kind of dissipating to the environment around it. And axons are exactly the same way, in which we have that myelin surrounding them and kind of conserving the loss of any of those ions that are going to take away from the generation of further action potentials down that axon. So last time we kind of started to introduce the concept of the 5 steps of chemical neurotransmission. Someone asked, do you have a good mnemonic device to remember the 5 steps of neurotransmission? And my initial response to that is no. But with that being said, we've got a group of probably like 20 very smart brains in here. And our steps of neurotransmission are synthesis, storage, release, receptor, transmission. So we didn't do a kind of an open and getting to know you thing. So find a partner and come up with a possible mnemonic device for your 5 steps of neurotransmission. Okay? So that's what we're going to kick off and do here as our first kind of activity as part of a class. So find a partner, come up with a mnemonic device, and I will ask a few of you to share them out if they're particularly clever. So find a partner, come up with a mnemonic device. Ready, go.
I first saw like soon. And after which feels like I'm like, you know, moss pit. But, you know. I don't know, my first day. Yeah, no, it's just. Yeah, the first letter never did my brain. I'm just. Oh, no. I haven't come up with the word spell. So, Jen, a little bit out there tonight.
Did you guys come up with anything good? No. No? Back of the room? Oh, Miles, I'm excited to hear this. Not Miles. Oh, Mitchell. Your name's supposed to start with an M. I don't know how that happened. Okay, Mitchell.
Snakes squeeze rats really tightly.
Oh my gosh. We must, you know, what's funny is that's almost exactly what I was coming up with when I was looking. I was like, slippery snakes regurgitate rats tomorrow. What a rat. Yeah. Anybody else? Oh, that's a good one. Yeah. Anyone else want to share a possible mnemonic device? Okay. You're like, no. Okay. Well, again, it's not a bad exercise to go through. When you have these stepwise processes, sometimes mnemonic devices can be kind of helpful and just kind of help lay the foundation. So again, synthesis, storage, release, receptor binding, and transmitter inactivation. And even some of those terms probably are kind of up for debate, but we will talk about them further today. Okay, so step one is going to be biosynthesis. So biosynthesis of the neurotransmitter in the presynaptic terminal. Now, as we spoke about last time, many neurotransmitters require some type of amino acid precursor. And that amino acid precursor gets transported into the cell. And then once it's in the cell, it gets kind of decorated with additional functional groups that make it a neurotransmitter, that make it a signaling molecule. So we have, we'll go through the stepwise process for glutamate, GABA. This morning I lectured about dopamine. For example, the precursor amino acid for dopamine is phenylalanine, for example, and then you undergo a series of enzymatic reactions that convert that to a neurotransmitter that is able to signal. Often, again, a prerequisite for a neuron being considered a neuron of a specific neurotransmitter subtype is it has to have the enzymes present to convert it into that final signaling molecule. They should be present in that presynaptic neuron. Their cofactors should be present in the appropriate compartment, and the synthetic pathways for neurotransmitter formation have long been targeted as an important action for clinically useful drugs. Okay, so this first step again is synthesis. You got to make your signaling molecule. Second step is you have to package your signaling molecule. So to package your signaling molecule, you do so into a vesicle. A vesicle is not that different from maybe like a micelle in the fact that you have kind of your extracellular components that are able to fuse and exocytose the neurotransmitter into the extracellular space. Last time I spent a little bit more time discussing the differences between neurotransmitters and neuropeptides. Does anybody remember how these things are different? How are neurotransmitters different than neuropeptides? Are they different? Maybe that's the question. Yeah, come on, speak up, you got it. Neuropeptides are proteins, exactly. Neuropeptides are proteins. They're actually string of amino acids. So they are much larger molecules than our neurotransmitters are, which are just slightly modified amino acids. Because of that, the types of vesicles that they get packaged into are also different. One question I think I saw in here, and I'll answer it now, is that neurotransmitters tend to signal into what we call point-to-point neurotransmission, typically, not always, but typically, where you have one presynaptic cell releasing the neurotransmitter and then a single kind of postsynaptic cell that's targeted by said neurotransmitter that expresses those receptors. Neuropeptides, being much larger, tend to have an increased level of stability and often can act in ways that hormones act, in which they are released from areas of our brain like the pituitary, which is a subset of our hypothalamus, and then it's able to signal across the rest of our body, right? We talked about vasopressin earlier this semester. That would be considered an example of a neuropeptide, and you have vasopressin receptors kind of distributed all across your body. So neuropeptides, larger, more structurally stable, and can signal vast areas across our bodies. And so their ability to signal between kind of our central nervous system and our peripheral nervous system is one of the ways in which we can really cause giant kind of effects in our body. Okay, so we have neuropeptides. We have Neurotransmitters, they're both getting stored in vesicles. So again, first step is synthesis, second step is storage, third step is release, okay? So how does a neurotransmitter, or why and how does a neurotransmitter get released? What has to happen? What's gonna cause neurotransmitter to get released? Or does it just happen spontaneously? No? Any hypotheses? So you're not necessarily going to see like receptor binding per se. When I say release, I'm talking about the fusion of the synaptic vesicle to exocytosed presynaptic compartment. So you're not necessarily binding receptors per se at that point. Yes.
This is with the snare complex.
This is when the snare complex comes into play. So for synaptic release, what's going to happen is you have your vesicle, and I always think of my vesicle as almost like a hot air balloon that has those tethers or those ropes that hang down, and I think of those ropes are kind of like those snare proteins that interact with calcium. Now, how does calcium get involved here? So if we talk about the traveling of an action potential down an axon, once it gets to the presynaptic compartment, we will have an influx of calcium through voltage-gated calcium channels. This isn't new information, correct? Yeah. You have calcium fluxing in through voltage-gated calcium channels, and those molecules of calcium are going to interact with the snare complex and essentially facilitate the release of neurotransmitter into the synapse. Neurons can use two different pathways to secrete proteins. The release of most neurotransmitters occur by a regulated pathway and controlled by extracellular signals like the influx of calcium. So what does this actually look like? So here we can see some examples of the snare proteins. This feels a little bit like cell biology to me, but it totally is. In the case of the top panel there that you see A, we have our vesicle, we have it, a little protein hanging outside of it called synaptobrevenin, and then you have syntaxin that's tethered to the plasma membrane. And like I said before, I always literally think of these things as the ropes hanging you down from a hot air balloon that somebody's grabbing and kind of twisting together. And that's really kind of what happens in this case, is you have these snare and snap proteins that are going to form this complex and basically pull the vesicle down to have it fuse and exocytose into the extracellular space, okay? Snare and snap proteins, super sensitive to calcium. Calcium influx is what facilitates this fusion process. And so it's the influx of calcium through those voltage-gated calcium channels that allows this to happen, okay? Questions?
Does it go all the way to priming before calcium?
Yes, it sometimes can depend on the synapse, but usually you do have a certain degree, a certain number of prime vesicles that are just kind of sitting there waiting for calcium to flux in for that kind of last step to happen before you get full exocytosis and release. And keep in mind, I'm trying to teach this like this is the way it always is. The probability of release or the probability of synaptic vesicle fusion can actually vary between neurons as well. And that's something you can calculate using electrophysiology. We will go totally down that rabbit hole. Yes.
Is it, I might be remembering wrong, but there are enzymes and whatnot that can like modify certain parts of the snare complex where it won't go through. Is that true? Like there could be.
Yeah, all of these snare complex proteins are proteins, right? So if you phosphorylate them, you can obviously alter things like their active site. So yes, there probably are enzymes that can modify this release process through that mechanism of modification. Other fun fact, oh, go ahead.
Do the vesicles just and diffuse towards the membrane and like wait there until calcium comes in, or is there another function that pushes the pump there?
So, I'm trying to think of like a great example. So like cholinergic synapses, for example, they have almost like two pools of vesicles. They have what they call their readily releasable pool. The readily releasable pool is going to be the pool that is like basically primed and ready to go. And then you actually have almost like backup synaptic vesicles that are going to be kind of hanging up there to replenish the readily releasable pool. These two pools of like vesicles that we're discussing here, most often seen in like the neuromuscular junction synapse, just because when you have an action potential traveling down an axon into your musculature, you want to make sure that you have neurotransmitter kind of primed and ready to release there, especially when you're undergoing kind of consistent activation of a specific set of musculature. So I'm thinking about like somebody running a marathon, right? Every time that person takes a step, they need to have acetylcholine ready to go. And so it's very hard to, what's the word I'm looking for? You to be able to essentially utilize all of the primed synaptic vesicles. So yes, it depends on the synapse again. You have yours that are primed here. There is a specific transport process that's going to facilitate them into both the tethering and the pairing process. So you have proteins that are kind of consistently bringing those synaptic vesicles down to kind of get them ready to release at the level of the synapse. Okay. The last thing that I'll mention to this is the snare proteins, interestingly enough, are a target of a bacterial toxin that you've probably heard of. Botulinum neurotoxin or Botox, for example. The way that Botox works, which again, Botox is literally just botulinum neurotoxin that is like taken from Clostridium botulinum. And the way that it works is it actually cleaves these snare and snap proteins. It actually cuts them so you can't have synaptic vesicle fusion that occurs. So when somebody gets Botox injected into the musculature of their face, for example, it stops this vesicle fusion from happening so they can no longer contract the muscles of their face. You've stopped that process from happening. Now, we utilize Botox for, again, removal of things like wrinkles. You can use it for pain mitigation as well. But if it, again, is consumed in a high enough concentration in food, that's how botulism is essentially contracted. And in the case of botulism, sorry, this is something I used to study, so you could probably get me to go on about this for hours. But botulism is interesting, and the botulism toxin is interesting because it most quickly intoxicates synapses that are under very rapid use. So usually the place that you see intoxication happen with a fully intact individual is their diaphragm. So they undergo like respiratory, because your diaphragm is a part of your musculature that is always working to help you breathe. So you see pretty rapid intoxication, and it's that inability to breathe that usually is what causes mortality related to botulism. But if you're put onto a respirator early enough in that intoxication process, you can probably be made to survive until those snare and snap proteins get remade. But you have to kind of wait for that process to happen in that meantime. So I studied botulinum intoxication for the Army. Don't be scared, but but if you have questions about that, I'm happy to answer them. Yeah, Paris.
Could Botox be used to like stop like uncontrollable muscle spasms then? There's like a medical use.
Yeah. So it's used, like you're saying, to stop muscle spasms. It's used in musculature. Some people have found it helpful for things like migraines as well. So there definitely is like a clinical use for Botox, but I think it's more widely known for like, dermatological reasons. But yeah, it definitely has a clinical use too.
Also fast degrade to move it ever.
Yeah, it does degrade. So eventually your physiology will break down the toxin and it will stop the cleavage process on these snare and snap proteins. I would roughly guess like 3 to 4 weeks is my estimate, but don't take that one to the bank. It's been a long time since I've looked at that literature. And I think that they're actually trying to modify versions of Botox so it does last longer, which is, I don't know if that's terrifying or helpful, but if you're using it again for a clinical purpose, it's probably great if you can extend and stop the fusion in the neuromuscular junction. So, okay. So step 3 here again is going to be release. So synthesis, storage, release, and next we have the post the release. So you have a vesicle that's fused, you have neurotransmitter that has been released into the synaptic space. Keep in mind, when we're talking about the synapse, you have your presynaptic cell where your neurotransmitter is getting released, and then you have your postsynaptic cell that has receptors expressed on it that neurotransmitter is going to be binding. The space between those two cells is the synapse. Okay. Now, at that point in time, you have the neurotransmitter has the capacity to bind to the receptors that are expressed at said synapse. Now, we've talked about ionotropic and metabotropic receptors previously, so hopefully this doesn't feel like too much of an intellectual lift, but here you can see the glutamate molecule, and again, it's just a modified amino acid binding to either ionotropic or metabotropic G protein coupled receptors. For glutamate specifically, which is our primary excitatory neurotransmitter, it can bind to either AMPA or NMDA receptors. Those are the currents that are produced by each of those respective receptors. And when we go into our glutamate section, we'll talk about how and why these two receptor, their currents look so different from one another. And then, so ionotropic receptors would be the fast signaling sets of receptors, but glutamate also can bind to metabotropic receptors or G protein coupled receptors that are coupled to G proteins. Metabotropic glutamate receptors are grouped into 3 different groups based upon their G protein coupling. Now, most neurotransmitters that are of like the GABA and glutamate subtype, since they're the primary kind of brake pedal and gas pedal, glutamate being the gas pedal, and then GABA being the brake pedal, they have both ionotropic and glutamate and metabotropic glutamate receptors, or GABA receptors, I guess. Now, once we get into talking about things like dopamine and serotonin, for the most part, those are all going to be coupled to G protein coupled receptors. Okay. So the next step is binding to these receptors. We usually think of neurotransmitter receptors as being localized to the postsynaptic neuron. So this is the example. We're looking at a presynaptic and postsynaptic neuron. In this case, we're kind of looking at maybe the dopaminergic system or even the serotonergic system. So this particular cell is releasing those neurotransmitters. And keep in mind, we think of our receptors being on kind of the receiving postsynaptic neuron, but you can have presynaptic receptors that act as autoreceptors. The function of autoreceptors is almost like a negative feedback loop system that we build into the synapse, right? So if you, let's say you release a ton of dopamine, and the dopamine is hitting your postsynaptic D1 and D2 receptors, but you do have a subset of D2 receptors that are expressed presynaptically. And once those get activated, those are coupled to G proteins that are inhibitory. Okay, so D2 receptors are coupled to G alpha I and O, which is going to inhibit further release of neurotransmitter like dopamine in this instance. So this is kind of like a built-in organizational technique that our cells have to make sure that you just don't continuously release like a ton of, let's say, dopamine. These again are the function of autoreceptors. They function as homeostatic feedback that modulates the function of presynaptic neurons, regulating transmitter release or synthesis or the firing rate of the neurons. Okay. Now, presynaptic neuron, postsynaptic neuron, you've got your autoreceptors. Okay, I'm doing a lot of hand gesturing, which means it's probably time for me to draw this. Okay. This is your presynaptic neuron. Here is your post. Okay, so this is kind of what we consider to be the prototypical signaling. Now, if this is released and it hits here, this is your autoreceptor. And for most intents purposes, we talk about this signaling from this neuron to this neuron, but we can also have circumstances in which this neurotransmitter escapes and hits maybe like a neighboring neuron. And that is considered a heteroreceptor. Yeah, heteroreceptor. Okay, so this is when you have neurotransmitter that hits adjacent receptors on a different cell. Does that make sense? So this is considered like homosynaptic neurotransmission. This is heterosynaptic transmission. And again, we've got our other mechanism here, which is autoreceptor functioning. Questions?
Specific reasons why it might like not go strictly to that one that we're thinking of, or it just happens?
So, okay, that's a great question. And I'm going to answer it probably in too much depth, but just, so close your ears if you don't want to hear this part. So, so when we're talking about homoreceptor activation, right, this neuron is trying to send a message to this neuron, and you want this interaction essentially to strengthen. Now, when you have, let's say, a heteroreceptor, often where those heteroreceptors are going to be activated is in the area that we call the perisynaptic region. And the perisynaptic region is kind of like this, we'll say like the sides of the neuron. It's not like exactly the postsynaptic density. And the types of receptors that get activated here might be of a different type. So if they get activated by this, it weakens their interaction because of the subtype of receptor that it's hitting. Does that help?
Yeah.
So the reason that it might hit heteroreceptors is to just kind of further strengthen this interaction and dampen down any possibility of this neuron kind of talking to this neuron because of the receptor subtype that it's hit. Now, again, if we're gonna, this would be like maybe an NR2B receptor of the NMDA receptor. That's kind of the perisynaptically expressed receptor that's very sensitive to glutamate. So if that's hit, this neuron will be like, I don't want to talk to you, but this neuron will get hit straight on, and you'll have AMPA receptors and NMDA receptors being hit here, and they're going to be like, this is awesome. Keep talking to me loud and proud. Okay. So that's kind of the function for the heteroreceptor, is it kind of helps, the short answer to this is it helps with synapse specificity. Yeah. Yeah.
I was reading a paper, about, just like cannabinoid receptors and whatnot,
Mm,
and the CD1R being a presynaptic.
Mhm.
So it goes, and then it goes back to presynaptic.
Mhm.
Yeah, I was just gonna say, I think it's a really good example of, I have a whole paper about this, what I'm trying to say. Of a really good example.
Yeah. Okay. You guys want to learn about cannabinoids really quickly?
Yeah, it's really cool.
You're like, yeah, let's talk about that instead of this. This is way more interesting. Okay.
It's super.
Okay, so what we're talking about now is in terms of synaptic transmission is like the most prototypical type of neurotransmission that like 9 out of 10 neurotransmitters, this is how they act. Is they're released from the presynaptic cell and bind to the postsynaptic receptor. Now, cannabinoids, specifically endogenous cannabinoids, were only really well characterized in the last 10 to 15 years because they are so, for lack of a better term, weird. Like they do things that no other neurotransmitter system that we know really well does. So, so in this case, I'll talk about a system that I know pretty well because I've published on it. But but if you can have a cholinergic neuron, for example, like muscarinic receptor subtype 1 or 3, acetylcholine's coming down and binding that. Now, the interesting thing about endogenous cannabinoids is they're actually lipids. Okay, they're lipids, and they can actually be cleaved from the postsynaptic neuron, and those lipids come up and bind to usually CB1 because we're talking about the brain. CB2 exists in the brain as well, but it's far more found in like immunological cells in the periphery. And so this particular type of signaling system, it acts in what we call a retrograde manner. You have retrograde signaling, and cannabinoid receptors are G protein coupled receptors that are coupled to G alpha I and O. So they're inhibitory, which based off of what you know about what cannabinoids do to kind of anyone with a central nervous system, they tend to cause cognitive depression. And it's through this mechanism of, again, hitting a G protein coupled receptor on a presynaptic neuron, it's lowering down the release of further neurotransmitter. The other interesting thing about cannabinoids is that they are broken down via enzymes, not taken up by transporters. So that's interesting. There are some hypothetical transporters out there though, but for the most part, the way we teach about them is they're broken down by enzymes like FAW. I think that's fatty acid amino hydrolase. But there's a couple different enzymes that break it down, which again is pretty unique that we're not using primarily transporters, but we have these enzymes breaking it down. So yeah, cannabinoids.
I can send that paper.
Yeah.
Yeah, I'll send it to her.
Woo. Oh, and I still need to share that other paper with the group about Hodgkin and Huxley. Okay. Okay. Other questions? Directly or indirectly related to content? I will tell you this, like, asking questions not related to exactly what's on the slide is what this class becomes. because eventually when second semester rolls around, there's going to be so many kind of fun and interesting side roads we can take. That's probably one of the most fun and interesting parts of this. So, okay, so we have our steps of neurotransmission. I'm going to blow by the next slide because I think it's misplaced. Boop. And the last step here is we have synthesized, we have stored, we have released, we have, bound to our receptors. And the last step of this is to inactivate and terminate the action of the released neurotransmitter. So if you have continuous activation of a neuron by a presynaptic input, does not necessarily convey dynamic information. Neurons are kind of prickly things in the fact that if you continuously release neurotransmitter onto a receptor, that receptor will eventually undergo what we call desensitization and downregulation. It's like, I've had too much signaling for today. I'm going back into my endocytosis like little vesicle. Now, those receptors can get recycled back to the surface, but again, that process definitely happens within those neuronal interactions. So sustained signaling has adverse effects. Examples include tetanus in muscles or seizure discharges in neurons. That's when we have kind of unregulated signaling that occurs in those instances. Now, there are multiple processes to terminate signaling. The first and most common that we'll discuss in here is going to be reuptake of the transmitter through membrane transport proteins. They're most commonly found in kind of the presynaptic neuron. They also can be expressed on the astrocytes that surround the synapse as well. These are two very common locations of reuptake. So here we can see a plasma membrane transporter shown on the postsynaptic cell and a glial cell express transporter. If we're talking about glutamate, for example, those are called excitatory amino acid transporters, or EATs. And a second active means of terminating the action of neurotransmitter is by enzymatic degradation. Okay, we just talked about the cannabinoid system. That is one system that uses enzymes to degrade their signaling molecule. The other, does anybody have a hypothesis as to the other neurotransmitter that gets degraded by an enzyme and not taken up by a transporter? I don't know if we've explicitly stated in this class, so you might have to pull from a different class. But the other one would be acetylcholine. Okay, acetylcholine is the predominant neurotransmitter in our peripheral synapses and our neuromuscular junctions. The enzyme that breaks down acetylcholine in those synapses is called acetylcholinesterase. Okay, it breaks down acetylcholine and stops that signaling. So, like I said, 9 times out of 10, you have these little neurotransmitter vacuums soaking up neurotransmitter. And kind of that 1 time out of 10, you have enzymes like acetylcholinesterase breaking down acetylcholine. Diffusion of the transmitter away from the transmitter receptors in the synapse is a passive mechanism that can stop neuronal signaling, but you might at that point in time also have it hitting your heteroreceptors at other cells as well. Okay. Questions? Oh, we are 41 minutes in here, and have we stood it all? Okay, let's stand. Let's stand and stretch our legs so we can finish out this lecture strong. Okay, stand, find somebody that you haven't talked to yet and share with them either what your superhero name would be or what your superhuman power would be. You can choose which one, because I think we've done superpower before. But what would be your super name?
Oh, right, yeah, yeah, no, I know. I just. That's all right. Oh. I'm saying.
What's that? Yeah. Okay. Yeah, sure. It's good, it's good. It's like, it's got. It's good.
Yeah, that's my favorite. Yeah, you should.
Hmm.
I love.
Okay. Yeah. Okay. Yeah. Okay. Yeah. Yeah. So I yeah. Yeah, so. Read on your water. So that's why you need your. Yeah. Yes, that's, okay.
What the what? Oh my God.
Like I used to also.
Yeah. That's pretty good. That's it.
I think.
Okay, let's go ahead and find our seats. Find our seats. Oh. Okay. Okay. So, okay, so the next 5 slides are slides that have way too many words, and I'm just going to preface this with that. But the 5 slides are representing if you were a benchtop researcher and you had a hypothetical neurotransmitter that you were trying to characterize, what are the steps that you would take to identify that neurotransmitter and validate it as a possible, like, real come to life neurotransmitter? Now, the purpose of these slides, again, is not to overwhelm you with words, but to kind of show you like how neuroscientists still think of the 5 steps of neurotransmission, even when we're trying to characterize a new and novel signaling molecule. Okay, so how do we define a neurotransmitter? the first criterion that you'd have to fulfill is to define a compound as that must be synthesized by and released from neurons. So you have to find the enzymes that are creating this biosynthetic pathway to lead to the release of this signaling molecule from the presynaptic cell. So again, this is almost following the 5 steps of neurotransmission kind of side by side. First, synthesis. Are the enzymes present? Second, it should be released from the nerve terminals in a chemically or pharmacologically identifiable form. Meaning that if you took a bunch of those neurons and you stimulated them and they released vesicles, you should be able to take that extracellular liquid and kind of basically run it through an HPLC and say, this is a signaling molecule that I find within this solution, okay? One should be able to isolate the transmitter and characterize its chemical structure. Again, that would be kind of a mass spec HPLC kind of situation that would be happening there. Third, a neurotransmitter should reproduce at the postsynaptic cell the same effects that are observed after stimulation of the presynaptic neuron. Meaning, again, if you are doing this kind of in a lab type situation, when you stimulate a presynaptic cell and you put a recording electrode in your postsynaptic cell, you're going to get some type of fun squiggly line. Might look like this, might look like this, who knows? It could look like any of these different varieties of channel opening situations. Now, when you isolate out your signaling molecule, and let's say you just puff it onto a neuron, if you puff it onto the neuron, you should get the same effect as if you stimulated the presynaptic neuron. The concentrations of the applied transmitter should be the same as those measured after the stimulation elicited release of the neurotransmitter. This is when you get really into the weeds of like receptor kinetics and how you can saturate the receptor and cause different types of responses. So you just need to make sure your concentration is the same, whether it's being released from that presynaptic neuron or you're exogenously applying it. Fourth, the punitive neurotransmitter should be blocked by known competitive antagonists in a dose-dependent manner, and the treatments that disrupt the synthesis of the candidate transmitter should block the effects of presynaptic stimulation. Okay, so this is where pharmacology comes into play. Okay, so if we are talking about using a competitive antagonist, have we defined these terms previously? Yay or nay? Getting Is that the response? Okay. So we're going to draw our synapse in a slightly different manner. Here's our presynaptic cell. Here's our postsynaptic cell. Here is our receptor. Okay. Now, when our neurotransmitter comes down and binds this receptor, let's say it binds right at this very position here. Okay. A competitive antagonist would compete for the exact same site, and the competitive antagonist would come, bind there, and essentially block our endogenous ligand or neurotransmitter from being able to bind that same site. That's what we're saying when we're saying a known competitive antagonist. An antagonist binds to the same site that this would, and it basically blocks it out from being able to have access to it. Make sense? Okay. So lastly, there should be appropriate mechanisms to terminate the action of the neurotransmitter, looking at enzymatic degradation and reuptake of the substance. So basically, if you did have a new and novel neurotransmitter that you needed to do step-by-step experimentation to validate it, these are the sets of steps that you would do. And interestingly enough, they very much kind of walk right alongside those 5 steps of neurotransmission. How do we measure neurotransmission? Well, this is an example. We are able to actually poke electrodes into our presynaptic neuron and our postsynaptic neuron, and we can measure the action potential as it travels down the neuron and how it's received by our postsynaptic neuron here. So we have a series of electrodes that are represented in this figure here. This is showing us kind of the relative recordings from each of those places. And then here you can see some of the downstream recordings. I'm not going to get too caught up in this process, but again, it's important. Okay, how can we target pharmacologically in these 2 processes of synaptic transmission? So the 5 steps of neurotransmission, again, synthesis occurring here, storage occurring here, release occurring here, binding occurring at the receptor point, and then termination, which is, again, in this case, the transporter. This is the dopaminergic neuron synapse. We have synthesis of, again, our presynaptic dietary phenylalanine coming in, getting converted to tyrosine. That's also being converted further into dopamine. So here we can see a drug that can block this biosynthesis step here. So step 1, synthesis. This is a drug that can block the synthesis of dopamine. Here we have the storage component, which is step 2. The storage of dopamine into a vesicle is facilitated by a protein called VMAT, and you can block that by a drug named reserpine. Release. What's a drug or toxin that we talked about that can block release? Botox or botulinum neurotoxin, yeah. So that's going to block our release step of this. Once you have synaptic dopamine, it can bind to our postsynaptic dopaminergic receptors or our presynaptic D2 autoreceptor. And the last step is going to be the reuptake process, which actually cocaine blocks the reuptake of dopamine into the presynaptic neuron. Thus, one of the reasons it is so addictive is because dopamine generally gets released to reinforce certain behaviors that are advantageously helpful or evolutionarily advantageous for us. And in this case, cocaine blocks that and keeps that dopamine hanging out in the synapse. Thus, it's like, yes, that's great. Do it more. Whatever you're doing, do it again. Yes.
MDMA, it essentially causes like a super intense efflux of serotonin, so much so that there's really like not like the withdrawals of MDMA is essentially like there's no serotonin left in your brain.
Mhm.
You have to like go through a process to reuptake that, not like resynthesize that. Is it because of like how does that work on a molecular level? Like how does it block any binding, or is it just simply like do an excessive efflux and that's like what's happening? I know that you can also MDMA can become pretty addictive. Yeah, over time if you use it too much. Yeah.
Yeah, so in terms of what's happening on the molecular scale with MDMA, most likely what you're seeing is a blocking of reuptake of serotonin, but then you're also, I think, inhibiting the enzymatic breakdown intracellularly. So here we have like our monoamine oxidase inhibitors can act to inhibit monoamine oxidase. So I assume it's like a combination of blocking transport, blocking degradation, and in some cases, maybe even facilitating further release. That's probably how you get that full just like release of those of all those serotonin vesicles at the same time.
Okay.
Yeah. When we talk about the synapse, just keep in mind again, we're talking about presynaptic and postsynaptic cells. And in terms of what the synaptic vesicles really look like under electron microscopy, here you can see exactly what synapses really look like. Each of these like kind of small balls that you see here, these are all kind of loaded up, and this is the difference between kind of a presynaptic and a postsynaptic cell, as you can see here. Okay, so let's kind of again go through the respective proteins that are helping this process. Okay, so let's talk about signaling in the central nervous system. So a few weeks ago, we kind of started our discussion with ion channels, right? Ion channels are what is going to facilitate the action potential of traveling. They're also going to be gating the influx of sodium, the efflux of potassium, and the influx of calcium. Keep in mind, again, this is kind of a nice review, is with ion channels, we can have voltage-gated ion channels, we can have ligand-gated ion channels, aka receptors, and then we can also have signaling gated ion channels. This is just kind of a nice review again to point out how important ion channels are to the synaptic signaling process and how they can also be targets as well. If we go from ion channels next to our receptors, we have both ionotropic and metabotropic receptors. Ionotropic receptors are really just ligand-gated ion channels. The ligand in that instance is going to be the neurotransmitter that it binds to. Your metabotropic receptors, or G protein coupled receptors, are 7 transmembrane regions that signal through second messengers. And this is again the difference between these two is you have fast neurotransmission and slow neurotransmission. Which of these two receptor types facilitates the flow of neurotransmitter into the cell? turn to somebody sitting next to you and have the discussion. Which of these receptor types facilitates the movement of neurotransmitter into the cell?
The line shifts. And the neurotransmitter is but in itself, it's cheap. Like the neurotransmitter. Yeah. No. No.
Okay, does anybody want to venture a guess here? Which of the receptors facilitates the movement of the neurotransmitter across the membrane? Anybody want to call me on it? Because it's a trick question.
Trick question.
It's a trick question.
I knew it.
You knew it.
Dude.
So keep in mind, the function of a neurotransmitter is to serve as a signal. The neurotransmitter itself does not move across the membrane. Receptors do not do that. Transporters do.
Yes.
Transporters transport the neurotransmitter into the cell. But the function of the neurotransmitter itself is to function as a signaling molecule. So it binds to the extracellular area of the G protein coupled receptor, causing a structural change, causing those G proteins to essentially go do their signaling jobs inside the cell. And in the case of the ionotropic receptor, it binds to that ionotropic receptor, allows it to open, and allows for ions to move across the cell membrane. So in the case of this, sodium would be said ion. Okay, so I'm not saying this to be tricky or mean or whatever, but I know that it's a kind of a common misconception that I've bumped up against in this class. So I just want to make sure it's clear to you that the function of the neurotransmitter is as a signaling molecule to activate these receptors, and then it gets taken up into the cell via transporters. Yes.
They always take glial cells. You see that, but are they
No.
always taken into glial cells, or are they also transporters on the cell membrane?
Yes. The answer to all of your questions is yes, I think. You see a high level of expression of transporters on glial cells, and then you'll also see them expressed on pre and post synaptic cells as well. So for example, if we're talking about glutamate, the majority of glutamate uptake is facilitated by the EAT2 subtype of transporter that's found on the glial cell. And then we have like EAT3, EAT1, which can be found on the pre and post synaptic cells respectively. So those transporters are slightly different in their structure, ever so slightly different, but they're just expressed in different areas of the cells, but you have them expressed in all of those different places. So yeah. Okay, when we talk about neurotransmitter characterization, I think this is actually the second time we've seen this respective slide. And this, for me, is just sometimes nice to see the big picture of how neurotransmitters are related to one another. And these categories are based off of kind of the size and structure of the neurotransmitter. Acetylcholine is an ester in terms of its basic chemistry. I'm not showing you structures here. I personally do not love chemistry. I probably have told you that before, but you guys are like, I'm in it right now, and I don't. No. But sometimes understanding the structural relatedness is helpful. The amino acid neurotransmitters, glycine, GABA, glutamate, aspartate. Again, GABA and glutamate are going to be the two that we focus the most on. For the amines, we see catecholamines and indoleamines. Our catecholamines, epinephrine, norepinephrine, and dopamine. Fun fact about our catecholamines, epinephrine and norepinephrine are kind of primary neurotransmitters that signal in our autonomic nervous system. And dopamine, to a certain extent, at high enough concentrations, can actually bind to the receptors that epinephrine and norepinephrine bind to, specifically the ones on our heart. Indoleamines, things like serotonin, histamine, and taurine are, again, structurally related in that sense. And then these are examples of actually, it says peptides here, but these are all examples of neuropeptides that are a larger structure of a protein that are further cleaved down into structures like endorphins, enkephalins, and substance P. The last one over here, we can have signaling and binding via ATP, nitric oxide, carbon monoxide, but we won't be getting into that a ton. Okay. Lastly, we have our ion channels, we have our receptors. Lastly, we have our transporters. The second ago, somebody was asking about transporter expression. This is actually an example of kind of showing both neuronal transport and reuptake. We also have glial transport and reuptake on this figure too. Okay, so again, transporters assist in the termination of neurotransmission. The other method is through enzymatic breakdown. The example that we go back to there again and again is acetylcholine. That's what ACH stands for, is acetylcholine. And it's expressed on both neurons and astrocytes. It diffuses out of the synapse and is taken up via transporters. And many of the transporters are also co-transporters and utilize ionic gradients like, for example, sodium. Like the DAT transporter, the dopamine transporter, uses a sodium gradient to take dopamine back up and into the cell. How you guys doing? Good. Okay. The last thing that we're going to do today is kind of deviate from our prototypical chemical neurotransmission that we're used to talking about. 9 times out of 10, that's when we talk about neurotransmission. We're talking about chemical neurotransmission, in which there is a presynaptic vesicle, an active zone, postsynaptic receptors. You have a chemical neurotransmitter that's sending that signal. The synaptic delay is pretty significant because you have to wait for that diffusion of the neurotransmitter. And this generally just happens in a unidirectional way. I'm saying generally because there are exceptions to every biological rule, it feels like, and this being one of them. You can't have retrograde signaling, but again, that's the exception. We're contrasting this with electrical synapses, and the electrical synapses differ from chemical synapses. These synapses are a little bit more, I would say, simplistic in the fact that you just have 2 proteins butting right up against one another and sending ions right through them. They kind of take out the middleman of the neurotransmitter altogether. So gap junctions are what these synapses are essentially called, and they mediate electrical signals by kind of directly connecting the 2 walls. Here you can see the presynaptic and the postsynaptic cell, and this structure that you see here are actually 2 proteins that are so butted up so closely to one another that ions can directly flux from one cell to the next. They do produce ionic change in the postsynaptic cell. Yeah, and we can see here electrical synaptic signaling. The presynaptic terminal may contain ion channels. Postsynaptic cell must be relatively small for this to function. In this case, we're looking at kind of an experimental setup of an electrical synapse. You have your postsynaptic neuron here, and this is the giant motor fiber in which the interaction is occurring. The small cell has a high, what we call input resistance, so a greater voltage should change in response to the current. And so again, this is an example of kind of 2 neurons that are side by side that have essentially created electrical synapses between the 2. So if you cause an action potential to happen in cell 1, cell 2 will have enough ions that flux in to also have an action potential occur in that cell as well. These, interestingly enough, electrical synapses have also demonstrated both long-term potentiation and long-term depression. This is what another, just I think a more interesting maybe gap junction image. There is these, again, butting up of these 2 proteins that are embedded into those walls. Even a weak subthreshold current can depolarize or hyperpolarize the adjacent cell, and usually they are kind of very highly tightly packed in terms of distribution of those proteins. The proteins themselves, this is where biology actually helps you out with its nomenclature, they're called connexons. They literally just connect 2 cells, and they're called connexons. Is the name of that particular protein. Okay. Yeah, connected by hemichannels called connexons. There's generally these 1.5 nanometer pores, 4 alpha helices that span the membrane and close in response to lowered pH or elevated calcium, depending on it. Neurotransmitters from neighboring cells can affect the signaling in these, just depending on how it's altering that function. If we take a zoom in into the connexons themselves, here you can see again the presynaptic membrane. You have the protein that is a transmembrane protein, and your postsynaptic membrane, again, that has those transmembrane proteins. And you basically created like this nice little pathway or bridge between the neurons to cause function. Why is this type of synapse even found? What function do you think this might serve? No function? You're like, I don't know why this exists. Maybe this is like an evolutionary holdover from simpler systems. Possibly. Synchronous signaling. So when you need two cells to synchronously fire with a high degree of fidelity, this is when you see these electrical synapses take place, okay? There's one area of our thalamus in which you can find these electrical synapses, but you also find them in your cardiac musculature cells as well, right? It helps facilitate the synchronous contraction of your heart, okay? So electrical synapses, when we study them in other animal species, here's an example of speed being really important, the escape response of aplysia. Aplysia, as you can see here, small cells can act and coordinate as one large cell. So in this case, you have a sensory neuron that becomes activated. You have these motor neurons that are going to synchronously cause release onto the ink gland to cause one kind of ink fusion event that occurs within the aplysia species. So this is the motor response to the tail stimulation. And here you can see how synchronously those action potentials are fired amongst kind of these three motor neurons that you're recording from here. So when synchronous release of something is important, this is one method that we can utilize, I guess, to kind of get that synchronous release to happen. Did it. Boom. Okay, that's it. Questions? Your brain's full? Yeah. Okay, we did cover a lot yesterday, or I'm sorry, on Tuesday and today. Things that I would walk away thinking about or kind of, or mulling over is just like tying together the action potential, tying together summation, thinking about the steps of neurotransmitter release and the proteins that are facilitating them. As I said before, I consider this to be one of the maybe like most important weeks that we'll have in the semester, because every subsequent week, those 5 steps of neurotransmission, now we're going to go into each of the neurotransmitter systems and talk about what are the enzymes that synthesize glutamate, what vesicular transporter loads it into that vesicle, what the release is usually the same across, what are the receptor subtypes, and what are the transport mechanisms. So this is kind of, I would say, like the blueprint for all of the rest of the lectures. If you understand or have a nice strong foundation in those 5 steps of neurotransmission, then when we go back into the respective systems, you can say, okay, this looks a little bit familiar to me, okay? Okay, on Tuesday, come with questions if you have questions related to the exam. I'll be happy to answer those. I'll address some of the questions that you guys left me on Tuesday. I'll have a Kahoot ready for you. I will have your short answers questions written so we can go over those in class. So like I said, if you have questions between now and then, feel free to reach out. Does anybody need anything? Yes. I don't think there is, but I can post one. Yeah, I'll post a practice exam for no other reason than it keeps me honest and it makes me write a new exam every year. So I will just post last year's exam because I think that will be the one closest that follows like this year's blueprint. Okay, cool. Thanks for asking. Okay, good luck. Have a good weekend. Stay safe. Take care of yourselves. Make sure you're drinking enough water.